Today we're performing the DNA extraction lab using split peas, and DNA stands for deoxyribonucleic acid. DNA is a molecule that stores the genetic information needed to build and maintain an organism. So in step 1, I added salt, which was sodium chloride, and the salt helps the DNA separate from proteins and other cellular components and helps neutralize the negative charges on DNA. And this allows the DNA molecule to come together more easily during extraction. Step 6, I'm adding detergent because it's important to break down the cell membranes and nuclear membranes, and these membranes are mainly made of lipids, and the detergent dissolves those lipids and allows the DNA stored inside the cells and nuclei to be released into the solution. After the solution has cooled, I'm transferring it into the test tube until it is about 1/3 full. Now I'm adding about half of the papain powder, which is a meat tenderizer, and papain is a protease enzyme, meaning that it breaks down proteins and it helps remove proteins that are associated with or mixed in with the DNA, and this makes it easier to isolate the DNA. I'll then gently stir the papain into the solution so that I don't break up the DNA. At step 14, I'm using ice cold 91% isopropyl alcohol. I'm holding the test tube at approximately a 45-degree angle and slowly adding the chilled alcohol until there's about equal amounts of alcohol in pea solution. And the purpose of this chilled alcohol is to precipitate the DNA. DNA is not very soluble in cold alcohol, so when the alcohol is added, the DNA comes out of the aqueous solution and becomes visible. And I've provided some pictures and a video. At step 16, this is when we can see the results, and I can see the interface between the two layers, since this is where the DNA begins to precipitate. The extracted pea DNA appears as white, cloudy, stringy, or thread-like material at the boundary between the alcohol and the pea solution. My DNA yield seemed to be a lot more than I expected. The DNA was kind of a white, cloudy material, and it appeared pretty thick. And overall, I observed just much more DNA than I expected to see.